Hi guys, this is Mr. Arnold. We're going to talk a little bit about the principles of inheritance. So in our last video we talked about meiosis, which is how we create gametes, how we create sex cells, and that of course is the process by which we're going to pass on our traits. So we're going to talk a little bit today about the actual mechanisms by which those traits get passed on. So all of this starts with a guy named Gregor Mendel. Now, Gregor Mendel's kind of a sad fact because he's a monk back in the 19th century, so like slightly after post-Civil War era, I want to say 1866 or so. And so he's a monk, and at the same time he has this great interest in biology and particularly in pea plants. And so he's growing all of these pea plants. He keeps these extremely meticulous journals and does all of these experiments with them, and he's really the guy that comes up with the basics for even modern genetics. The sad thing is he didn't really get any attention paid to him at the time because he was a monk and he was sort of out in the middle of nowhere. But once biology really came to the forefront in the 20th century, Mendel's ideas get revisited and they sort of form the basis for all the stuff that we really are going to use in genetics now. So inheritance is the idea that traits are passed from the parents to their offspring. And so Mendel discovers this by investigating pea plants. And so there's 7 things that he checks out for them. So he checks out the colors of the seeds, he checks out whether the seeds are round or wrinkled, the flower color, what the pod looks like, how tall the stems are, all of these things. These are traits that Mendel is going to test a lot. So the first thing that he does is he makes sure that he has purebred plants. And what purebred means is that he breeds them for several generations that they always come out the same. And so we're going to look first here at the flower color. And so he breeds them for multiple generations so that he's only getting purple flowers, okay, in one case, and then in another case he's breeding the same flowers with each other and getting only white flowers, okay? So then what he decides to do is he takes a purple plant and a white plant, okay, purple flower and a white flower plant, and he crossbreeds them, okay? And so those are his parent generation, a purple flower and a white flower, and what he finds is that everything in the first generation after that, what we call the F1 generation, or the first filial generation, everything in the first filial generation is a purple plant. Okay, so that sort of leads him to say, okay, well, purple must be what we're later going to call dominant over the white flower because he didn't get any white flowers out. Okay, so when he crossed the first two, he gets nothing but purple plants. So here's what's interesting though. What he then does is he takes 2 of these F1 plants. So let's say he takes these 2 right here, and he crosses them and gets a second generation, or what we call the F2 generation. What he gets then is pretty interesting. He gets essentially, and this is a ratio, okay? They're not getting exactly 4 plants. What he's actually getting is he gets 8,000 plants. And what he finds is that he gets around 6,000 or so are purple flowers, and then around 2,000 or so are the white flowers. Okay? And he does this for all those 7 traits that we talked about and takes these really meticulous records. And again, we're not talking he did it once and found out what happened and that was it. He does it thousands and thousands of times. Okay, and records the data and says this is the probability of these things. So what he's saying is that the probability when he crossed 2 of these F1 plants together, that statistically speaking, he was going to get about 75% purple plants and 25% white flower plants at the end. So what conclusions does he draw, and what does this mean for us today? Well, the first thing he draws is that there has to be 2 forms of whatever leads to the flower color. In other words, there can't just be one thing that says flower is purple or white, because if that was the case, then how does he get the white flower here in that F2 generation? There has to be at least 2 things there. Okay, today we call these things alleles for a particular gene. So we've got a trait like flower color or like seed pod color or maybe eye color or something like that. And there's a particular gene that codes for that, but there's 2 copies of that gene, and each of those is called an allele. Okay? Now, this leads to what we call the law of segregation, which says that those 2 alleles are going to separate when we do meiosis. Because remember, we've got 2 copies normally, and then in meiosis, you get copied so that you get 4, it gets split up, and then each cell has 2, and then it gets split a second time so that each cell ends up with 1 copy, one of the alleles. Okay, so let's see what that looks like. Remember we said that he concluded that the purple plants were dominant, okay, and that the white plants must be what he called, what he termed, or what we're going to term now, actually, sorry, as recessive. So dominant means that it overrules the recessive. You only see the recessive if that's all there is. And so what do we mean by those two alleles? Well, this is what we mean here, is that you've got two copies. So let's say that the color here is coded by a P, and capital P means the dominant purple, and the lowercase p means the recessive white. And so you got two copies of each. We could have two capital P's, definitely going to be purple. We could have a capital P and a lowercase p, that's still going to be purple because purple is dominant. Or we could get two lowercase p's, in which case that's going to be a white organism. Okay. Now there's some terms here. If we only have if we only have one type of allele, so they're either both capital or both. Okay, so homozygous means that they both have the exact same letter. So lowercase p, uppercase p, those are homozygous. Okay? So it's heterozygous, or what we call hybrid, if there are two different letters. And in that case, the color is determined by which one is dominant. In our case here, capital P means that the purple is dominant, and so that's what we see. Okay? So a couple of good terms there you need to know: dominant and recessive, and then homozygous and heterozygous. Make sure that you got those terms. That also leads us into two other really important terms. These two other terms are the genotype and the phenotype. So the phenotype is essentially, what we can say there is, okay, what does it look like? Okay, what do we see? Okay, like Mendel isn't looking at the DNA. They don't even know about DNA when Mendel's doing the experiments. What do they actually see? You see the phenotype. The genotype is, what are the actual alleles? What are the letters that get split up? So capital P, lowercase p, both of these genotypes here, okay, the homozygous dominant and the heterozygous, both lead into purple plants. To get the, to get the white plant, okay, I have to have homozygous recessive. Okay, so genotype is the letters, phenotype is what it actually looks like. Now, what does that mean? We said that there was a term called segregation. Okay, so what does that whole segregation thing mean? So this is what it means. If we've got two hybrid parents, okay, hybrid meaning heterozygous, so they each have a capital P and a lowercase p, then when we cross them, what happens is that the letters actually split up. Okay, so one gamete gets an uppercase P, another gamete gets a lowercase p. Okay, so let's say that's one parent, and then the second parent, the same thing happens. They split up. Okay, we arrange these into this little box structure here called a Punnett square, and then to see what the children, what the offspring are going to look like, we essentially just carry everything down. So if I've got an uppercase P here, okay, then I go over here to my Punnett square and I say, okay, so I'm going to write, or it would help if I had the pen on. So I'm going to write uppercase P, that's an uppercase, okay. Down the bottom, I write another uppercase P. Over here, I write a lowercase p and then another lowercase p. Okay, so that's me carrying these traits down. Okay, and then I'm going to carry these traits across. Okay, so I'm going to write those. So this is an uppercase P, so we're going to write that. Okay, that should be an uppercase P. And then this again is an uppercase P. Why? Because I'm carrying this straight across. Down here, lowercase and lowercase, okay, because we're carrying this straight across. Now, what does that mean? Well, what that means is that now I've got a homozygous dominant, definitely purple, right? Okay, so homozygous dominant, definitely purple. And then I've got two hybrids, and we know that the hybrids are purple because purple is dominant, okay, so those show up. And then we've got are homozygous recessive, where they're both lowercase, and that gives us a white pea plant. So what we see in the phenotype then, sorry guys, me and the pen are having issues here. That's what happens when I record at school. So what we have for the phenotype, okay, of this cross is we've got 3 purple, okay, and 1 white. Now, we could also break that down and we could write the genotype, although it's unlikely that you're going to be asked that very often, but if you are, the genotype for this would be 2 capitals, okay, I've got 1 of those, and then the hybrids, I've got 2 of those, and then the recessives, I've got 1 of those, okay? So what they look like, and then what their letters actually are. All of that is done by Punnett squares. And we're going to work some Punnett squares in class. I actually think, really, once you get the hang of it, Punnett squares are kind of fun to do, and it's kind of fun to figure out what this stuff is there at the end. So the other big conclusion Mendel has as he does a lot of this stuff is that he's breeding for a bunch of these traits at once, right? He's not always doing just one. And what he finds is that the traits sort out separately from each other, okay? In other words, if he did a color and a height, that he could have short white plants or tall white plants, or he could have tall purple plants and short purple plants, that those things weren't tied together. It wasn't like all of the white plants had to be short also, or all of the tall plants had to be purple, that those things separated out independently from each other, okay? And he found that by doing what we call a dihybrid cross. So a lot of wall of text here. I'm sorry about that, but we've got it all separated out. Hopefully the colors help a little bit. And so this is what we're going to do. We're going to look at a dihybrid cross to see how this all works. So first off, flower color, this is what we've been doing so far. So capital P is purple, lowercase p is white, purple's dominant, white's recessive. We're also going to throw in now the color of the P. And so in the color of the P, yellow is actually dominant and green is recessive. So that means capital Y is yellow, and that's dominant, and lowercase y means green, okay? So if we had a dihybrid, meaning for both traits, so the P's here are for the flower color, and then the Y's here are for the color of the actual P, okay? So purple, yellow. Now, this would be a purple flower, yellow pea plant, but it's a dihybrid. So the phenotype is purple flower, yellow pea. The genotype is capital P, lowercase p, capital Y, lowercase y. Okay? So how does all that work out? Now, you are not going to have to perform, we might work a couple in class for fun, but on a test, you're not going to have to perform a dihybrid cross, but you need to know the law of independent assortment. So here's what it means. It means that out of that dihybrid phenotype that I had, okay, capital P, lowercase p, capital Y, lowercase y, that I can separate that out into different genes. So every time I make a separate sex cell, a gamete, that what I do is I pull one from each of these. So one P and one Y. Okay, that's that segregation part. Remember, they split in half. So the P split in half and the Y split in half. I could also have a capital P and a lowercase y. I could have a lowercase p and a capital Y. I could have a lowercase p and a lowercase y. That is what we call the law of independent assortment, saying that if I've got two traits, okay, each with two alleles, that those things can all separate out. And so out of those two traits that are dihybrid, I've got four possible gametes that I could get out of this. And that's what's important here, is to realize that what we've got down here on the bottom, it's not just a bunch of letters. These are the traits that are going to be in the gamete. So that's a gamete, and that's and that is a separate gamete. Sorry guys, me and my pen today are having problems. Okay, and then another gamete here. So we've got four sex cells here. Okay, so each of those things separate out. Now, if we were doing a dihybrid cross where each parent was that dihybrid, then that's what we would do here, and we would write this out. And so I would have that capital P, capital Y, okay, capital P, lowercase y, lowercase p, capital Y, lowercase p, lowercase y. Okay, and then we would do the same thing. If we're doing a dihybrid cross and they were both dihybrids, then we would put that here as well. Okay, come down to the next one. We've got that same P and Y, lowercase p, capital Y. Make that a little clearer. And then we come down here to the bottom. We've got lowercase p and lowercase y. And then we would carry the things across and down, just like we did in our other cross. Okay, so I'll show you how that works out, but don't stress real hard about how it works out. In fact, what I'm going to do is I'm going to take everything from the top and I'm going to label it. Let's see if the green will show, or the blue here will show up. So I'm going to come down with the blues and then I'm going to come across with the yellows so that you can actually see how this works out. So everything gets a big P on the way down. Okay, and they get a capital Y. Now I'm going to leave a space because I want to keep my P's together and my Y's together, so I'm going to leave space for that second P that I get from the side. Okay, so that's why I'm leaving that space there. Don't let that panic you or anything like that. So that's one column. And then just to keep it simple, I'm just going to carry across the yellows over here from the other side. I'm not going to work the whole thing out because that would take a little while. Capital P, uppercase Y, and if we would keep carrying that across, right? P, Y, P, Y, big P, big Y, right? And so what we would get here just from this first offspring here is obviously we're going to get another purple flower and yellow pea because we've got nothing but dominance. If we went down here to the bottom and we cross this, and I'll just go ahead and bring that down so you can see that. So little p, little y. Let me switch back to my blue real quick. And then down here at the bottom we've got capital P. I'm sorry, whoops. That's not capital P, right? Because if I'm bringing it down from the top, it's little p, right? And that's what I wanted to put there. So little p and little y. Okay? So in this case, double homozygous recessive, and so I'd get, in this case, I'd get the white flower and the green peas, which is going to be the most rare. If we filled in the whole thing, and I'll show you a copy of this in class, if we filled in the whole thing, we would have a bunch of different phenotypes and genotypes here because we're talking about two traits at once. Okay? Don't let that dihybrid cross throw you off. We'll probably work some in class, but you're not going to be tested generally on dihybrids. What you're going to need to do is to be able to do a single hybrid or just a 4-box Punnett square, which is pretty simple. We'll work through a bunch of those. All right? Thanks for listening, guys. Hopefully that gives you a clue on how Mendelian genetics work and how to set up your Punnett squares.